Hello everyone, my name is Lance Ramsey and I'm the marketing manager here at Formulatrix. I want to begin by thanking everyone for taking the time to join us today. So welcome to our protein crystallization automation webinar series. Before we begin, I'd like to go over the agenda for this first session. First you'll hear from Mr. Sudarshan Murthy at the University of Oulu. Then we'll hear from Dr. Yael Pazy Benhar from the Technion Center for Structural Biology at the TCSB. During the presentations, please feel free to enter any questions you have into the GoToWebinar interface. With that, I would like to introduce our first speaker from the University of Oulu, Mr. Sudarshan Murthy. And sir, I'm going to pass over the controls for you. All right, sir, the floor is yours.
Thank you, Lance, for the introduction and stuff. So, good morning to the people joining from the Americas, and then good afternoon to people joining from Europe. I'm Sudarshan Murthy. I'm a Ph.D. student in University of Oulu, Finland, and today I'll be talking about crystallographic binding studies of ADP-ribosyltransferases using the Instruct and BCO core facility in Finland. Yeah, to tell you briefly about my project, I'm working on a human depleted toxin like ADP-ribosyltransferases. So these are enzymes which catalyze a post-translational modification, that is ADP-ribosylation. So what happens here in this enzyme uses NAD as a cofactor, and it cleaves NAD, and nicotinamide is released, and ADP-ribose is added to the target protein, in turn changing the properties of the target protein. So in humans, there are about 17 enzymes of this family, and then they're classified into 3 different groups based on the catalytic activity. So there are poly-ARTDs, mono-ARTDs, and one of the enzymes, the activity is still unknown. So the poly-ARTDs are enzymes which add a chain of ADP-ribose to target protein. And then mono-ARTDs as the name suggests, that they're enzymes which add just one ADP-ribose to target. My project basically focuses on mono ADP-ribosyltransferases. So the objectives of my thesis are to develop potent and selective inhibitors for specific MARTDs, which can be used as tools to evaluate the cellular functions and also elucidate the concepts of ADP-ribosylation signaling. And this inhibitors can also be used as lead candidates for drug development against human diseases. So one of such mono-ARTD is the ARTD10. Why do we have to study ARTD10 is because it has been shown that ARTD10 is overexpressed in cancerous cells, and the overexpression of ARTD10 causes apoptosis like cell death, and it interacts with the PCNA, and it also sensitizes cells with DNA damage. So ARTD10 has also been shown recently to be involved in severe developmental delay. So deficiency of ARTD10 has been shown that there is a lot of developmental delays and also DNA repair defects. So as any inhibitor project goes on, we did start screening a lot of libraries for this against this enzyme, and one of such library was the NCI library, and through that we found a hit compound that was what we named as compound 1 here, and it had a nanomolar potency towards ARTD10, and it was also very selective against the other members of the family. So we wanted to study more about this compound and wanted to see how substrate relationship of the compound works and how to make this compound much better. So we use a traditional method by modifying the compound in different sites and then see how it works. So with that, we did a lot of modifications. We synthesized some of the analogs, we bought some of the analogs commercially, and then through these studies we found that there's one more compound which is called compound 32 here. It has a slightly better potency than the parent compound, and also it showed good selectivity towards ARTD10 than the other members of the family. These results were then later published in the European Journal of Medicinal Chemistry. So as my project encompasses a lot of structure-based drug discovery, I have to do a lot of crystallization trials, as you know. So for those trials, I use a lot of formulatrix images, and for this I use my core facility, which is available in Oulu, Finland. So here we have 3 formulatrix images, one RI54 image and the room temperature is 20 degrees, RI54 at 4 degrees, and then we have an RI2 at 4 degrees. We have approximately about 30 users from 6 different groups here, and approximately we produce about 1100 plates per annum. And all these images are then taken from the images and are imported into the ice bear for scoring and monitoring. I'll be talking about ice bear in the coming slides. So what is ice bear? Ice bear is a crystallographic image information management and research tool. It was developed by Ed Daniels, University of Oulu, in collaboration with protein crystallographers in Finland. So the purpose of ice bear is education, research, and infrastructure. Education, it makes it easier for the supervisors and also the students to monitor their experiments. And for research, all the information is then stored into the database, which can then easily be retrieved while we are writing manuscripts. And for infrastructure, as such excellent and expensive infrastructure which we have, it also requires very good, excellent IT infrastructure. So ice bear acts in such a way. So tell you about how ice bear looks. The login page looks quite simple. So here you have different bricks which you can customize according to your wishes, and the question mark sign here is the help button, which helps you navigate to this page. It's quite easy. Okay, so once I have set up my plate and then put it into the formulatrix image and get back to my computer, what I do is then I assign the plate to a particular project. So here it was ADP-10, or also called as PAP-10. So I assign the plate to a particular project. And then for this project, of course, we have, as we know, we have a lot of different constructs which we are trying simultaneously. So you can also add different constructs to that particular project. This has also other options, like you can, of course, go and optimize your drop conditions. You can go and optimize. You can also check your crystals, and you can mark them for taking you to the synchrotron trips in the later phases. Okay, so this is an example picture of how a drop viewer looks like. So here you have, if you can see at the bottom, you have different options like a tick sign, a clock sign, information sign, a drop sign, stuff like that. So each of the sign gives you different details about your experiment, and the I sign here is giving you details about the protein conditions and the plate, the type, and such like. All this information is available in that I sign. And then so then there is this crystal selection sign, which is a drop sign. So here you can select your crystal, and also you can search your plates and DVARs in the search, find your barcode symbol. To tell you briefly about what's exactly happening in ice bear that is behind the screens is you have formulatrix imager. All your images are then imported directly into ice bear, and ice bear have different modules. That is the drop view module, which I showed you earlier, and then there's this fishing module, and then there's the shipment module. So in the fishing module, what happens is basically you are connecting your crystal to your pin and your puck, and the puck to different position and the different positions in your puck. And then there's the shipment module. The shipment module is where you connect all the information, all the data from your ice bear is directly synced to your ice-PyB. And this is work in progress, which we are trying to do it, which is happening as we talk now. But so they are trying to make it an automated one. So as soon as you have your information fished your crystal, it automatically goes into ice-PyB. How does freezing of the crystal happen? So I use a barcode reader to scan my pins, pucks, and DVAR. And then I just use a mouse or a touch screen to just drag it through. So I can show you how it looks. Yeah. So the fishing module looks something like this. So you have crystals here, and then you have different pins which you have already scanned and kept. You have your puck which you have scanned, and the divas which are at the bottom. So select your crystal which you are sending and to assign to which pin it is in. So just drag it there and put it in the pin, and drag pin to the puck and puck to the diva. So it's as it is. Once that is done, you enter the shipment module. Here you have information about your crystallization experiment and also your diffraction experiment. This is an example slide for specifically for DLS, which we are working in collaboration with. So the QR sign here, QR code here, is also a clickable QR. So if you click on that, you get information of your crystallization conditions on your left-hand side. And if you click on where there is diamond light source QR, if you click on that, you get all your diffraction details. Once that is done, you have all your information and stuff. So the other major important task which iceberg does is what happens when you get back your diva. You either keep your crystal or you can wash it. Like some of them tend to, like if they didn't have enough time during this synchrotron beam trip, they would like to recollect their crystal elsewhere. So they can keep the crystal here. But if you don't want, you can just wash it. If you have already collected data, just wash it. So even this module has been useful in SBAC. Coming to the summary slide, as you know, as I told you earlier, chemical probe would help us study the roles of each of the enzymes and also help us to validate new drugs within the ATT family. So efforts are now ongoing to develop and expanding this arts to the other mono-ATTs as well. As of now, I have, for the MARTD project, I have around approximately 120 plates, and then I've marked about approximately 200 crystals for this project. So without the help of formulatrix images, this would have been a tedious task to do manually. This is my final slide. So I'd like to thank all the BCO core facility members for this. And then if you need more information regarding iceberg, kindly contact Ed Daniels and Rick Beringer. There's also a demo information here. You can click on the link and then use the username and password as demo and plate number 9098. You can just try out how iceberg works. Thank you.
All right, thank you, sir, for that presentation. Next, we have Dr. Pazy Benhar from the TCSB in Israel.
Hi, my name is Yael, and I'm the head of the Technion Center for Structural Biology, the TCSB. We're located in Haifa, Israel, and I'd like to tell you a little bit about our facility today and what we do with the formulatrix equipment. So first of all, this is my website, and this is the Technion Center for Structural Biology. This is our team, Dikla and me. I'm the head of the unit and Dikla is the lab manager. Our goal at the TCSB is basically to take proteins from solution and solve their three-dimensional structure. We have various projects coming from both academia, the Technion, other institutes and industry, including the medical school at the Technion. And we have various projects that are coming from labs that are, some are crystallographers, some have no structural background at all, and therefore there's a different level of assistance we give to different projects. So just to give you an idea why people are interested in coming to us, sometimes they have a protein they work on or an idea of biological problem, and they are interested in solving the structure for either understanding better how the protein works and studying the active site of the protein. Some laboratories are interested in the interactions of their protein with other proteins, and therefore they'd like to solve complexes of protein. And some labs are interested in drug design and engineering a specific drug for their protein, and they would all come to us to solve the structure. At the TCSB, we have 4 subunits, and I will briefly explain about each one of them. Since we solve structures at our facility with X-ray crystallography, Our main unit is our crystallization unit. We have a screening equipment, the Mosquito by TPP, which help us easily dispense buffers, precipitants, and the protein at very small amounts. We normally use 150 nanoliters, and it enables us to do high throughput screening of various conditions. It gives us high accuracy and reproducibility. From there on, we have various equipment from Formulatrix, which we are extremely happy with. The Formulatrix imager, for instance, allows us to automatically image and capture high-quality images of the different crystallization conditions. It's much better than old times when we needed to remember what was in each one of the drops. We can just look and see the growth of crystals over time, and our users can log in to the website and use the RockMaker software in order to detect crystals and also to see and mark their successful drops. It's extremely helpful to catch the exact time when the crystals have evolved inside the drop. We also have, besides the Rock Imager and the RockMaker software, which are extremely helpful and can help us easily screen the images, and we can also prepare optimizations with the RockMaker. We have the formulator optimization, which is also very, very helpful because we can use, it's super easy to prepare and fast to prepare optimization screens directly into the plates. And so we can use 16 different solutions and create either a grid plate or a random plate. And we have daily examples where you can see that we get from not so very good crystals, much better crystals using the optimizer, the formulator. So I'd say this is pretty much the main unit at the TCSB. Besides that, we have the heart of the unit where we have an X-ray generator by Rigaco, the X-ray system. Once we have suitable crystals, which sometimes we have to enlarge using seeding and crystal handling techniques, we load either manually or with our robot where it can automatically mount crystals on our FRX system. And here you can see our system. This is just a different image, but we have the FRX generator. We get diffraction, and you can see we control both the experiment conditions and the robot from a control room. You can see our robot in action. It can auto-mount crystals, takes them from the liquid nitrogen, loads them on the microglide, and we can center the beam on the crystal. And again, this is really helpful having the Formulatrix system close by. We can load crystals that evolved on that specific day when they're at their best and load them on the beam. Our third subunit is our graphics room, where we have several Mac systems and software to solve structures. This is the room where all the excitement happens when we solve structures. And finally, we have another subunit, our biophysical unit, where we have both an nano ITC and nano and MST and DSF from nanotemper, which allow us to both determine the stability of the protein and whether we have good binding and high affinity. So this is DSF. I will not explain much about it, but this is our biophysical unit. Many labs who are not interested in solving structures and just are interested in understanding whether the protein is stable or aggregated come and use our DSF. So basically, this is the TCSB, and I'd like to thank you very much for listening.
Well, thank you so much for that overview of what the TCSB has to offer. Just as a reminder, all the talks today have been recorded. You'll be able to find those on our website after session 2 concludes. And as a reminder, session 2 begins at 1:30 p.m. today. Finally, I'd like to thank everyone for taking the time to come out and listen to the talks today. Now with that, I would like to conclude session 1. And so I hope everyone has a great rest of your day. Thank you.